In this topic, we're going to discuss monoclonal antibodies. So we're going to look at what is a monoclonal antibody, and we're going to recap on B cells and antibody secretion, how are monoclonal antibodies produced, and then we're going to look at what are the uses of monoclonal antibodies. So I'm going to give you further information on the detection of prostate cancer, breast cancer treatment, and pregnancy testing. Now, when a pathogen, represented by this red circle, invades the body, this pathogen has got many different antigens on its surface, and each antigen will induce a different B cell to multiply and clone itself. So a mixture of different antibodies is produced. Now, in medicine, it's beneficial to produce a single type of antibody from just one clone rather than a mixture of them. These antibodies are known as monoclonal antibodies. So a monoclonal antibody is an antibody produced by a single clone of cells or cell line, and it consists of identical antibody molecules. Just to recap, immature B cells are made before birth, and it's when these cells mature that they have the ability to make antibodies. These B cells move into the blood and then they concentrate in the liver and spleen. When a B cell comes into contact with a pathogen, its cell surface receptors recognize the antigen on the pathogen surface, as you can see at the top there. The B cell then undergoes clonal proliferation, where some cells become plasma cells and others form memory cells. Those cells that become plasma cells secrete antibodies, and the antibodies from the plasma cells combine with the pathogen and destroy the pathogen. Now, although in this diagram it looks like those plasma cells are releasing their cell surface receptors, those in the last bit there, the pink represents the antibodies. The memory cells form the immunological memory, so that there's a faster immune response if the same pathogen invades the body later. Here's a structure of an antibody. So an antibody is made up of 4 polypeptide chains, as you can see on the left there. So you've got a pair of heavy chains and a pair of light chains. Then on the right, the variable region is where the antibody binds to the antigen. This region differs with each antibody. So it's a shape that fits exactly into that, or the antigen fits into that variable region. The rest of the antibody is the same in all antibodies, and it's known as the constant region. Okay, let's have a look at how monoclonal antibodies are produced. Firstly, a mouse is immunized with an antigen. This stimulates the B cells within the mouse to proliferate and to produce a mixture of antibodies. The plasma from the mouse is tested to confirm the presence of the antibodies. The plasma, or the spleen of the mouse, is then used to get the B cells that are making these antibodies. Now, to enable these B cells to divide outside the body, they need to be mixed with cells that divide readily outside the body, for example, cancer cells. So on the right here, you see the cancer cell line is going to be grown or is grown in tissue culture. These cells have lost the ability to make antibodies, and they're also modified to lack an enzyme for a particular pathway. Detergent, or polyethylene glycol, is added to the mixture to break down the cell surface membranes of both types of cells, so your B cells and your cancer cells, and this enables them to fuse together. These fused cells are called hybridoma cells. The hybridoma cells are separated out under a microscope. Now, the spleen cells and the myeloma cancer cells would have died, so the hybridoma cells are identified. Each single cell is then cloned, and each clone is tested to see if it's producing the required antibody. The group producing the required antibody is grown on a large scale, and the antibodies are extracted from the growing medium. Because these antibodies come from cells cloned from a single B cell, they are called monoclonal antibodies. These monoclonal antibodies come from mouse tissue, so they need to be modified to make them like human cells before they can be used. And we call this humanization. Okay, just a brief recap of what I've just said. The mouse is immunized with an antigen. This stimulates the B cells to proliferate and to produce antibodies. The plasma from the mouse is tested to confirm the presence of the antibodies. The plasma or the spleen of the mouse is used to get these B cells that are making the antibodies. The plasma cells are mixed with the cancer cells. Detergent is added to the mixture so that the cancer cells and the plasma cells fuse to form a hybridoma. The hybridomas that produce antibodies are identified and cloned, and then antibodies from a single clone are extracted and modified. Okay, let's have a look at some uses of monoclonal antibodies. Well, they're used in diagnosis, blood typing for transfusion, tissue typing for transfusion, diagnosing diseases, for example, the ELISA test for AIDS, identifying pathogens to distinguish between different strains, used to identify the location of tumors, testing for drugs, and testing for hormones, for example, the pregnancy test. Okay, a little bit of extra information. Looking at how monoclonal antibodies are used to diagnose prostate cancer. So monoclonal antibodies are very useful in the diagnosis of disease, for example, influenza, hepatitis, and chlamydia. They're also useful in diagnosing cancers, for example, prostate cancer. Now, with prostate cancer, there's a high level of the protein PSA, which stands for prostate-specific antigen. By using a monoclonal antibody that interacts with this antigen, the levels of PSA can be measured in the blood. So a high level of PSA can give a warning so that the person can go for more tests. Okay, treatment of diseases. Antibodies are very specific to particular proteins, antigens. Monoclonal antibodies can be used to target specific substances and specific cells. So one type of cell they can target is cancer cells. They can thus be used to treat cancer. The monoclonal antibodies are produced that are specific to the antigens on the cancer cells. These antibodies are given to the patient, and these antibodies attach themselves to the cancer cells. They then block the chemical signals that stimulate their uncontrolled growth. So an example is Herceptin, a monoclonal antibody used to treat breast cancer. Now, the advantage of direct monoclonal antibody therapy is that since the antibodies are not toxic, they lead to fewer side effects than other forms of therapy. So here you can see a breast cancer cell has got receptors, HER2 receptors on it. These all have pathways. If you add Herceptin, it'll block this pathway, so it stops the cell from growing. Okay, let's have a look at how monoclonal antibodies are used for pregnancy testing. Pregnancy tests rely on the fact that the placenta produces a hormone called human chorionic gonadotrophin, HCG. And this is excreted in the mother's urine. So let's have a look at how the test works. The test strip of a home pregnancy test kit is dipped into a urine sample. And this urine sample will move across the strip by capillary action. As the urine comes into contact with the membrane, it first mixes with antibodies linked to colored particles that are present in the nitrocellulose membrane. If HCG is present in the urine, it combines with the antibodies that are linked to the colored particles. The HCG antibody color complex moves along the strip until it reaches a narrow region where another type of monoclonal antibody is immobilized. The HCG antibody complex is trapped by these second antibodies, and this creates a colored line. So it's that first colored line. As the liquid continues to move towards the end of the strip, it meets a region that has another immobilized monoclonal antibody, and this one traps the colored particles whether they are attached to HCG or not. So the second line will appear for both positive and negative results, and this acts as a control. So if both lines are present, the test is positive. So here you can see a pregnancy test kit that shows positive for the lady being pregnant. Okay, we've looked at what a monoclonal antibody is. We discussed very briefly B cells and antibody secretion. We looked at how to produce monoclonal antibodies in a mouse. Then we discussed the different uses of monoclonal antibodies, and we looked at the detection of prostate cancer, so when levels of PSA are high, breast cancer treatment, for example, Herceptin, and pregnancy testing using human chorionic gonadotrophin. And that concludes our lesson. The end.